Right, hello and welcome back to the channel. It has been quite a while, it's probably been slightly over a week. I got hit down pretty badly by a cold, and then I had a week of work experience, so I've been extremely busy over the last few days, but I haven't forgotten about this. So today we'll be talking about digestion and absorption. Many people think that they're the same, but in fact they are two separate processes, which I will be explaining in this video. So before we dive right in, I would just like to say, if you do think this video will be interesting to you, then please make sure to like and subscribe, as there is a lot of other biological related content on my channel. So without further ado, let's dive right into the topic of digestion and absorption. So we've got the three. Okay, well, let's just define digestion first, and then we can relate that to the rest of the video. So digestion is the hydrolysis of larger biological molecules into smaller biological molecules so that they can be absorbed into the body. So what do I mean by hydrolyzed? Hydrolyzed is the breakdown of a condensation reaction by the introduction of a H2O water molecule, and that will break down a bond, which I will get onto in a second. There are many different types of bonds involved. That will break down that bond, making it into a smaller biological molecule so that you can digest it. Because when they're big, you can't, and I'll get onto that in a second. So, our first larger biological molecule that needs to be digested is starch. So this is done in a multiple step process, but basically, firstly the glycosidic bonds, which are basically starch is a polymer of alpha glucose, which has high glycosidic bonds between OH groups in the alpha glucoses. So yeah, in starch, the glycosidic bonds are hydrolyzed by amylase, which is produced in the pancreas and salivary glands, and this is done to release maltose. And in maltose, these glycosidic bonds, because glycosidic bonds are also in maltose to connect the two together, because maltose is a disaccharide. Glucose is a polysaccharide, but yeah, maltose is a disaccharide. Hydrolyzed by maltase, which is embedded in the epithelium of the ileum, which is in the small intestine. So yeah, and then that hydrolysis releases monomers of glucose. So now let's move on to protein. So in protein, there's these things called peptide bonds, and these peptide bonds are hydrolyzed uh, and for this specific enzyme, they're hydrolyzed in the middle of the polypeptide. And when they're hydrolyzed in the middle of the polypeptide, it's endopeptidase which does this, and endopeptidase is produced in the stomach/pancreas. This is to release shorter amino acid chains. Now, secondly, peptide bonds are hydrolyzed on the ends of the polypeptide chain by exopeptidase, which is released in the stomach and pancreas, and this is to release singular amino acids. So, peptide bonds are hydrolyzed between dipeptides by dipeptidases, which is found similar to maltase on the small intestine membrane in the ileum. It's embedded, and this releases single amino acids. So now before we move on to fat, you're probably thinking, why are these enzymes embedded into the small intestine rather than floating around? Well, it's because when it's embedded in the small intestine, they are not lost when excreted. So all these other digestional enzymes that are created in the pancreas or the stomach, they are lost, and then they have to be made again. So yeah, now let's move on to fat. Fat is when I'm speaking to fat, I'm mainly talking about triglycerides. That's going to be a glycerol molecule with three fatty acids bonded to it. So firstly, the bile acids emulsify the fat, and then the ester bonds, which are in the fat between the glycerol and the fatty acids, are broken down by lipase, which is created in the pancreas, and then this releases fatty acids and monoglycerols. You've got 2 fatty acids and 1 monoglycerol, because a monoglycerol is just a glycerol connected to a fatty acid. So that's per triglyceride. So now we will move on to absorption. Right, okay, so now we move on to absorption, which is the next part in this process, wherein by the products of the digestion of these larger biological molecules are absorbed through the small intestine and into the bloodstream, where they are used in the body. So, without further ado, let's move on to starch and protein first, which is glucose and amino acids, because they both follow the same steps. So, this right here, up here, is this. You can imagine this space up here to be the lumen of the small intestine, which is like, if you've heard of blood vessels, it's basically the space between like the walls of the small intestine. And then you've got the cells here, the cells of the ileum. They're ones, which is a part of the small intestine. They're 1 cell thick, so you've got this short diffusion pathway. And then out here is the bloodstream, so that's where it's being diffused to. So, for the first step is the active transport of sodium ions into the bloodstream, and this is via the hydrolysis of ATP, because during active transport it moves against the concentration gradient, so you require energy from ATP in order to have the energy to be able to perform that action. So the movement of sodium ions against the concentration gradients, and they're going from a low gradient to a high gradient. This maintains the concentration gradient of sodium ions between the lumen and the cell itself. So this means that sodium ions and amino acids both move in together in a process known as active transport, where you use an active transport or carrier protein there. And this is a part of facilitated diffusion. And then you've got the amino acids or glucose that move through the cell and then are facilitated diffusion out. This is either in a channel protein or a carrier protein, and it does not require energy from the hydrolysis of ATP because it is a passive transport. So that is how starch, which is hydrolyzed into glucose, and protein, which is hydrolyzed into amino acids, are transported out from the lumen of the small intestine through into the bloodstream, where they are then transported across the body and used for their specific functions. So yeah. Now fat, which goes to monoglycerides and fatty acids, that is a slightly more complex process. So basically, you've got your monoglycerides and your fatty acids, and then these join up with these things called bile salts, which I think are created in the gallbladder. I'm not entirely sure. And then that creates something called a micelle, and these micelles are basically bile salts and these monoglycerides and fatty acids all bundled together. Yeah, and these micelles are taken to the wall of the ileum in the small intestine, where their monoglycerides and fatty acids are released into the cell. And then in the cell, there's a thing called the smooth endoplasmic reticulum, which is used to convert these monoglycerides and fatty acids back into their triglycerides, so they're re-synthesized here, because that's what the that's what the smooth endoplasmic reticulum does. The rough endoplasmic reticulum has the ribosomes, and that does the protein, so the smooth does the lipids. So you've got your lipids that have been synthesized there, but the process is not completed just yet, because then you've got these go to the Golgi apparatus, which is another organelle which is inside the cell, and the Golgi apparatus mixes these triglycerides with some proteins, and this creates something called a uh, chylomicron. I will put that up there because that's hard to pronounce, but I think it's CH something and then cron. So yeah, sorry about that. And that's packaged into this little vesicle here, and then these chylomicron fuse with the membrane of the cell, and then they are then released into the rest of the bloodstream, which is where they are carried along to where they need to be and stored. So, without further ado, I'd just like to say thank you for watching this video. Sorry about my voice, it's probably been very sick, well not sick, it's probably been very, I don't know, difficult to listen to. So if you have got to the end of this about 10-minute video, then I would like to say thank you. It's been good to be back doing some videos. I hopefully will be doing a lot more. I just haven't been able to recently because of work, illness, all that kind of stuff. But yeah, digestion and absorption. If you've liked this one, go onto the channel, check out all the other ones that are there, and I will see you in the next one. Bye bye.